So I'm Professor Richard Burt, and I've written and succeeded in getting NIH grants, and so I'm used to reading NIH grants. And when COVID-19 came out, I stumbled upon some grants on the website that concerned me, and I did notify a major media news network that's international because I have spoken on it, and I have the name, and we have talked by texting back and forth. And they really weren't interested, and nothing happened, and we moved on with life. But I decided because people need to know, I wrote a book for the lay public. It's called Kill Switch: The History of How Viruses Shaped Humanity and Led to COVID-19. Now, I'm NIH trained, and I'm a professor that led a large immunology lab for a long period of time. So I can read grants. And I reference this book about 700 references, more like an academic book, so you can look it up. But it's really written for lay people, although I think some prior education would be helpful. But you'll really enjoy this. And I think it'll open your eyes. It's certainly opened mine. And that's why I'm grateful to have this opportunity to present it. So I'm talking about viruses, and there's a lot of people, especially in corporate media, that talked about COVID-19 for like 2 years, but I don't think they really know viruses or were trained in them. So I just want to ask the question, what came first, the chicken or the egg? Uh, so the answer to that is the virus or the viroid. The viroid's actually just a small single piece of RNA, whereas a virus is RNA and DNA with proteins, but they're very, very small. And that's what came first. So, and the reason I say that's what came first is because a virus or a viroid is a transition between the dead and the living, because part of its life, it's dead as a rock in that it has no metabolic activity. And that's when it's outside the cell. When it's in the cell, then there's metabolic activity, and new viruses, tens of thousands, are synthesized and released and often kill the cell they're in. So that's most likely, rather than getting stuck, you have to have a chicken to make an egg and an egg to make a chicken. We really go back to how life itself began. And it began probably with very simple viruses, most likely RNA, because RNA has a enzymatic function or ribozyme function in addition to being a blueprint or a code that is a historical record of the DNA or the nucleotide sequences. So a virus was the original inhabitant of this planet. That's a quote from the X-Files movie in 1999. And that's surprisingly somewhat relevant to this. Viruses may be RNA or DNA, and they contain from 1 to 100 genes. The RNA viruses tend to be simpler, really 1 to 10 genes usually, wrapped inside a shell that is a protein coat. And that's all they are. So viruses are metabolically inert or dead outside the body, but can return to viability or life after being outside the body for hours, like rhinoviruses that cause common cold, or days, like influenza or COVID-19, or months, like rotavirus or hepatitis, or years, like smallpox. Viruses are the largest biomass on this planet. So there are more viruses than there are any other type of bioorganism, including bacteria, fungi, insects, mammals, and humans. There are more viruses than everything else combined. And in fact, there was a paper in Science where if you look in human stool, there's about 1 billion viruses per ounce of human stool. So that's a bit unusual. It's like humans, by eating food, feed bacteria that feed viruses. So in an upside down twist to evolution, we may not be the top of evolution. You can ask the question, do we really exist as incubators for viruses that are the largest biomass on this planet? So the virus came first. An percent of our human DNA consists of remnants of old or ancient viruses, and another 40% is made up of repetitive strings of genetic nucleotides that are also thought to have a viral origin. That's not my quote. That's a quote from Cold Spring Harbor Laboratory. So roughly 50% of our own DNA are either remnants of old viruses or appear to be repetitive strings of these viral nucleotides. So we are descendants of viruses. And so you have to wonder sometimes, if we go around and play with the foundation of life, that is with viruses, which are the largest biomass on this planet. Could we affect ourselves in unanticipated ways? So the light microscope allowed us to see cells and bacteria, but not viruses. They're too small. You couldn't see a virus. So how did we know viruses exist? Well, I think that's an important question because one time I was giving a talk to a lay audience and someone spoke up, said, have you ever seen a virus with your eye? And so before I could answer how we know they exist, he was told, so you can't believe they exist. Well, how did we know they exist when we couldn't even see them with a microscope? So before we could see a virus, we knew they existed as something so small it could pass through a porcelain filter, which is made of a clay, a diatomaceous earth and water that was fired in a kiln and that could stop bacteria or cells. But what filtered through, the water that went through, could still infect and kill animals. And so they knew there was an infectious organism in there, and they knew it was smaller than anything could be seen with the microscope, smaller than bacteria. So they called it virus, which is Latin for poison. It's where we get our word virulent. And the person who came up with the porcelain filter was Louis Pasteur, the Pasteur Institute in Paris. But in the 1930s, Ernest Rutherford developed the electron microscope, and that allowed us to visualize viruses, for which he got a Nobel Prize. So to understand how COVID came about, I had to get the history of viruses, and these are the viruses talked about in Kill Switch, and the story behind each of them is really fascinating. It's a historical perspective, so it's very important to read it from the front to the back to appreciate that history wrapped around often the discoveries of scientists, especially Nobel laureates, that allowed us to understand and manipulate these viruses. And in the far right-hand column, I'm showing the three-dimensional structure as visualized by an electron microscope of viruses from smallpox down to COVID. So Kill Switch is a historical interaction of humanity with these viruses. Remember, a large part of our own DNA appears to be viral in origin. So they're not these, as our name says, poisons or virulent or virus from Latin that can kill us, but they also seem to play a very important part of who we are and in evolution, what we've become. So what is a vaccine? A vaccine's a virus. That is, it's a virus that is alive, but domesticated it. We domesticated this live virus, so it's much less virulent. And then when you're infected with it, you develop an immunity to it, but it doesn't cause you any significant disease. And I'll talk briefly in a little bit about how we domesticated these viruses. Or a vaccine may be a dead virus that is embalmed, like the Egyptians used to embalm mummies, so that the three-dimensional structure is still there, so your immune system can recognize it and attack it, but it's dead and it's not going to cause any disease in you. Or a vaccine may be a piece of a virus that is a protein or messenger RNA, as in the COVID vaccine, which is messenger RNA, or is in even papillomavirus, where it's just a piece of the virus, or an Ebola virus, where it's kind of a piece of a virus engineered into an entirely new virus that has never lived before on this planet. So a vaccine is a kill switch to prevent a pandemic. So basically, when I first decided to write about this book, I was talking with U.S. Senator Mark Kirk about it. And I said I was going to write it. And he said, yeah, I think you should title it Kill Switch. And I think that's a very appropriate title because there are many important kill switches that prevent good intentions from going bad. A virus through a vaccine is itself a kill switch to help prevent pandemics and, mass mortality of the human population. So here's the vaccines that were developed before 1960. That is the pre-molecular biology age. And you can see over, 160 years, there weren't very many vaccines developed. That's because, the tools to do it were very laborious. That is, either attenuating a live virus or embalming it and making it a structurally intact but dead virus. So the first vaccine, the very first ever, was a live smallpox virus that had a different route of entry. So normally the smallpox virus enters your respiratory system. It causes, it's transmitted through respiratory particles, so, by coughing, sneezing, or, through the respiratory fluids. And it causes painful vesicles in the skin and mucous membranes, the eyes, the respiratory tract, the GI tract. And this virus, because I wanted to show you they really exist and under EM what they look like. This is a double-stranded DNA virus that has this kind of dumbbell-shaped nuclear protein inner core. So the first vaccine was the live smallpox virus delivered to the skin, and that was called variolation, which is Latin for cutting. So the smallpox first became prevalent during the Roman Empire, and it killed about a third of Roman citizens and probably fundamentally changed that empire and set it into one of the first problems for decline. In fact, it was brought back to Rome by Roman legions after fighting in Mesopotamia. And, it decimated the Roman legions because they lived in close quarters. This empire ending virus resulted in not enough troops to defend the frontier. So there was a big push to hire German soldiers to work for the legions, but they had to defend against Germanic tribes trying to enter the empire. So, but it began in Rome, and one of the greatest, probably, emperors, the philosophical emperor Marcus Aurelius, who wrote Meditations, which is often still studied in philosophy classes. One of his famous quotes is, "The best revenge is not to be like him or her who hurt you." But anyway, this virus killed Marcus Aurelius, and it had a mortality about 15 to 30%. So why would anybody intentionally give them the live virus? Well, what happened is normally it was delivered by inhaling it into the respiratory tract, but the Romans quickly learned that if you cut it into a skin, so if you took one of these painful vesicles, put a knife in it, got it contaminated with the virus, and then just made 3 cuts in the skin of a person that never had smallpox, their mortality would only be 3%. It would drop from 15 to 30% to 3%, but then thereafter they would be immune to it and they couldn't get infected anymore. So it was your choice. You could take that 3% chance of death from variolation, that is cutting the virus into your skin, superficial cuts, or 15 to 30% chance if you acquired it naturally. It was your choice. But why would it work that way? The Romans didn't know. The Europeans in the Middle Ages didn't know. They know it worked, but they didn't know why. And the reason is when it enters the skin, it's about a week before it actually gets into the blood and towards the respiratory tract and starts causing symptoms. During that week, when it's still stuck in the skin, your immune system has time to respond. Not your innate, that reacts immediately, but your adaptive immune system. It gives it time to respond to develop antibodies to it. And that's why it markedly decreases the death rate. It's the same lethal virus, but it's how it's delivered that's important. So while the Romans had developed variolation, there's still a need for an effective vaccine that didn't have a 3% chance of killing you. And that was first developed by Edward Jenner in the UK in and that vaccine, he noticed that girls on farms that milked cows never got smallpox, and the cows had cowpox, and the girls didn't get cowpox. So he figured they got infected with a virus from the cows that really didn't hurt them, that gave them immunity to smallpox. So he started intentionally taking the pustules on cows or horses, horsepox or cowpox, and injecting them into people and then into children. And then he would inject them with smallpox, and they never got smallpox. They were immune. And so the second vaccine, or the first real vaccine, outside giving the live virus in a different way, like they did with smallpox, for smallpox, the first real vaccine was a live animal virus that was not pathogenic to humans, but conferred immunity to smallpox. And Edward Jenner was markedly criticized for doing this. It was called a cuckoo idea. And one of the reasons they called it a cuckoo idea is because another prior paper that Edward Jenner wrote was about the cuckoo bird. He was the first to observe that a cuckoo hatchling, that the cuckoo birds laid their eggs in another bird's nest. And the hatchling, when it was born, would kick the native hatchlings out of the nest. So the parents would raise the cuckoo bird as their own, not knowing it murdered its litter mates. And so they began to, he was the first to report that. So developing a vaccine, they called it a cuckoo idea, but it worked. And Jenner coined the word vaccine that we use today, and it comes from the Latin word vacca, which means cow, because he got it from cows. He took it out of the vesicles of cows, or vacas, and so he called it a vaccine. And the cowpox and horsepox was originally passaged in animals, so they'd always have a supply, and later, as we learned, in animal cells, so there would be a supply, and it remained the vaccine against smallpox until the last documented case of smallpox in Somalia in 1977. Since then, there's been no more smallpox. It's been removed from this planet. That virus no longer exists in nature. And it's the only virus to which a vaccine has totally eliminated the virus, which is good news. We can applaud that. But again, we also have to recognize that we're evolutionarily derived from viruses, and our genome is about 50% viral in origin. So could there be unintended or unexpected consequences of eliminating viruses? Totally. So the first dead virus, so we talked about the first live virus vaccine, now we talk about the first dead virus vaccine, and that was rabies. And I have a picture of rabies. They're bullet shaped. It's single-stranded RNA virus with 5 genes. Only 5 genes cause rabies. And that because they're negative stranded, they must be translated into positive RNA, that is messenger RNA. And it's done by one of the 5 genes in the rabies virus. That's an RNA-dependent RNA polymerase protein in the virus. Rabies is this bullet-shaped virus, and it's named from the Latin word rabere, meaning to rage. And that's because the symptoms are a frenzy, raging, excessive salivation out of the mouth, an agonizing thirst, yet a tremendous fear of water or hydrophobia. How does this hydrophobia come about? This fear of water, despite being so thirsty? What happens is one of the proteins from the rabies virus, remember there's only 5 genes, 5 proteins, but one of them hitchhikes onto the dynein motor protein complex. And the dynein motor protein is actually a protein that walks up an axon all the way back to the nucleus along microtubules. It actually moves and walks backward. It's like this minibus carrying things back to the nucleus. And axons can be extremely long, but dynein motor protein does that. It's a normal protein in a cell. But rabies attaches to that. It's carried back to the nucleus where it replicates, and then it travels back down the axon to enter the salivary glands and laryngeal muscles specifically. In the salivary glands, it causes the virus to be released into saliva, so it enters the mouth, and then in the laryngeal muscles by the nerves that innervate the laryngeal muscles. Whenever you swallow, they spasm, so you can't even breathe. So you get really thirsty, but you're scared to swallow because your throat spasms and you can't breathe. And that results in this fear of water because you see it, your throat will spasm and you can't breathe. So there's this salivation where you're dripping saliva like a rabid dog, but you can't swallow it. And so in terms of evolution, the virus makes sure it's not being swallowed back into the stomach where the acid would destroy it. Instead, it can only go out of the mouth and to allow you to bite someone else and to further infect another animal or human. So this is the most lethal virus there is. It's 100% fatal once symptoms start. And it's usually transmitted by a mammal bite, usually a dog or a bat in the Western world. And when I say it's 100% fatal, there's about 10 people in the world who survived once symptoms started. Only 10. Once symptoms start, it's almost for sure you're not going to survive, even if you get the vaccine after symptoms. It's too late. So Louis Pasteur, who invented the porcelain filter so they knew viruses actually exist and that they're smaller than bacteria, he invented this vaccine. And what he did is he took the rabies virus, injected it into rabbit spinal cords when they got rabies. He took the spinal cord and either embalmed it by formalin fixing it or even just let it sit at room temperature and become desiccated and very dried out. You'd have to do that for more than 6 to 7 days or the virus would still live and be lethal. But once it was dead, and usually then with formalin fixation, formalin cross-links proteins so that it kills the virus, but the three-dimensional structure is there. It's like embalming and that the injections did on a corpse. So you could mount an immune response. So it prevented rabies, but about 10% of the patients injected with the original Pasteur vaccine developed experimental allergic encephalomyelitis in the human patients. It's an MS-like disease, very fulminant. So, 10% fulminant MS-like disease is better than 100% dying, but they improved it. They realized that what was causing it is the mixture of the virus and which stimulates innate immunity, and proteins of myelin from this rabbit spinal cord that caused the immune response to shift towards attacking myelin as well. And recognizing that, this became a model that is still used today. I used to do it in my immunology lab all the time, where you can induce autoimmune disease in animals, and these autoimmune diseases are perfect mimics for MS or arthritis, collagen-induced arthritis, adjuvant arthritis, whatever, myasthenia gravis, lupus, etc. And then you can study an autoimmune disease by doing that. But the point is, you get it by using a vaccine and a self-epitope, and that was learned from the rabies vaccine. But still, 10% severe MS-like disease from this vaccine was unacceptable. And what was realized is once you removed the non-neural tissue, and in fact began to grow the virus in even non-neural cells, that prevented this MS disease after administration of the rabies virus. So who gets rabies? Well, animals, or who gets the vaccine? Animals are giving it, and humans in high-risk occupations such as veterinarians are vaccinated to prevent rabies. But the general population is not vaccinated. And as a result of eliminating the source, which is usually your pet, there's only about 3 cases per year in America. But there's 30 to 40,000 worldwide, and they're usually children. They often occur in what has traditionally been considered the third world, such as Asia or Africa, where they don't vaccinate their dogs or cats or pets. And if bitten by a suspected rabid animal, they don't have ready access to getting the rabies vaccine to prevent the infection. And it's a horrific death for children. So that was the first dead vaccine, the rabies. What's the first live attenuated, or if you will, domesticated virus? So it's just like, domesticating a wolf to make dogs, and we bring out the traits we want in different dogs. Well, you can do that with a virus. And the first one it was done with is the yellow fever vaccine. And that was grown in embryonated chicken eggs. That is, chicken eggs that had been fertilized, started to form a little embryo. And it is the only Nobel Prize for a vaccine. It was given to Max Tyler at the Rockefeller Institute in New York in 1951. So this yellow fever virus is a single positive stranded RNA. So it acts as one long messenger RNA with one open reading frame, that is one start and stop codon, but it encodes 10 different proteins through post-translational modifications. So flavus, this yellow fever is a flavus virus. That's Latin for yellow because it causes liver disease, jaundice, and death. The source of yellow fever was discovered due to the Spanish-American War that occurred in 1898. So what happened is the U.S. battleship Maine was in the harbor in Havana, and it just suddenly blew up. And the Spanish soldiers, because Cuba was a part of Spain at that time, jumped in rowboats, went out to try to rescue everybody they could, help everybody they could. The hospital doctors and nurses were 24/7 trying to save American sailors' lives. But, over 200 sailors vanished, died in that explosion. The captain of the Maine survived, and he telegraphed to Washington of the good intentions and help from the Spanish, and that a Spanish admiral was there wishing his best. Nevertheless, the newspapers started saying this was an act of sabotage, and they even coined the term, "Remember the Maine to hell with Spain." Now, why would they do that? It turned out that the newspapers were owned by 2 very powerful people, and they competed against each other. One was William Hearst. The movie Citizen Kane is about him. The other is Joseph Pulitzer, and Joseph Pulitzer established the Pulitzer Prize. But what they did is they practiced what's called yellow journalism, which is still practiced today. They knew that if you emotionally triggered your audience, many more people would buy their newspapers and make a lot more money. So it was in their interest to sell their newspapers to make a lot of money by stating that this was an act by the Spanish Empire to destroy an American warship. Nothing could be further from the truth. Spain was bankrupt at that time. America was a rising power. They didn't have the money to fight America. But the war started, and they, out of just their history of being the seventh largest empire on earth, fought back. But it only lasted about 4 months before they had to sue for peace. And in the peace agreement, they gave Cuba and the Philippines and several other pieces of their empire to America. That's how we got it to sue for peace, even though they didn't start this, and they actually tried to help them. And then later, Admiral Rickover, who's the father of our American nuclear navy, commissioned a study to find out what happened to the main, and it turned out it was a design fault in the ship, in that they put the coal bunker right next to the furnace. And it was very easy for a spark from the furnace to get into the coal, ignite it, and blow the ship up. And that's most likely what happened. It was an engineering design fault. It was never the Spanish that did it. But because of that, when the war happened, about 30,000 US sailors and soldiers died, and they mostly died of yellow fever. They didn't die of the Spanish killing them. So the U.S. Army put together the U.S. Army Yellow Fever Commission to find out why these soldiers died. What was the cause of yellow fever? That's what they wanted to know. That's the first thing you need to know to try to prevent it. So they sent Walter Reed, a major in the U.S. Army, as well as Jesse Lazar, James Carroll, and other people to Cuba. And the first person they ran into in Cuba was Carlos Finlay, an epidemiologist in Cuba. And he had already published that the cause was the mosquito, because he correlated the incidence of yellow fever with the number of mosquitoes that were there, every spring. And he had actually been given the name Mosquito Man and ridiculed for that correlation. Nobody believed him, but Walter Reed did. And so Jesse Lazare and James Carroll had themselves bitten by mosquitoes that were feeding on people with yellow fever. And both Jesse Lazare and James Carroll got yellow fever. Jesse Lazare died. James Carroll survived but suffered permanent heart injury, from which he died 2 years later. So they presented that data, and America had a meeting in Indiana, and they were ridiculed, saying the mosquito couldn't cause it. The Washington Post even wrote about it, saying this is the most ridiculous idea for the cause of yellow fever that's ever been put forward, and there's been a ton of trash put forward. That's basically what they said in a newspaper. So what Walter Reed did is he set up Camp Lazare in honor of Jesse Lazare, who had died. And what they did is in one building, you would be given the soiled linens of people who had yellow fever, in case this was carried by, feces content or sweat or whatever. In the other room, you were always given clean linens, but you were also given mosquitoes that had fed on people with yellow fever. And you could say, well, according to today's standards, that's unethical, but there was a consent given, and they were paid, if they got yellow fever, $500, if not at least $200. And that was in gold. So that was a lot of money back then. And the truth is, they had tons of people volunteering, because U.S. soldiers and sailors were stuck there until they got yellow fever before they could go home. The U.S. Army just stuck them there. So that was the person or the institution that will put these people at risk. And so the sailors and soldiers knew, look, either I'm stuck here till I get yellow fever and I die or get better and go home, or I do this experiment, and if I get it, I'm going to get a lot of money. And if I survive, I have the money. If I die, at least my family has that money, and we'd have the best medical care the Army could give us. So they had no problem with people wanting to do it. And given the constraints that they were in, it, I think, was a very ethical study. And they found that the mosquito caused it. And the first victim was actually developed yellow fever on Christmas Day. So that then led, by the way, the Walter Reed Medical Center in Washington, D.C., the premier medical center for the U.S. Army and Navy, is named after Walter Reed. Jesse Lazare has a stained glass window in the National Cathedral in his honor. In any event, this led the US military then saying, well, now we know the source is mosquitoes. And America then changed what it was doing. It would put oil over standing water to kill the mosquito larvae, and it used pesticide to kill mosquitoes, and that kind of prevented these yellow fever outbreaks in America. But they wanted to know the source, and it appeared the source was Africa, in ships coming over from Africa into the Caribbean and then to America. So the US Army sent the West African Commission to study the source. Two people in that commission were Dr. Noguchi. He's actually was Japanese. He had also discovered that syphilis, the syphilis spirochete, caused neurosyphilis, or insanity of the insane, as it was caused at that time. And William Young, they both died from yellow fever. What happened is Noguchi got it, yellow fever, and after died from it. William Young did the autopsy on him. On the autopsy, got cut and got yellow fever and died. But what this commission did is they established an animal model. That is, they realized that the yellow fever could be transferred from a person into a monkey, and then they could serially transfer in monkeys, and they'd have an animal model to study, which was really important and needed. And the human who gave the first yellow fever virus that they transferred in monkeys, by the way, these monkeys were from India that had never been exposed to yellow fever. African monkeys already had been, they wouldn't work, so they used Indian monkeys. But the person whose serum that got yellow fever was a SIBI. And so the strain of yellow fever vaccine that's still used today is from a SIBI, from the virus isolated in his blood. So the SIBI strain ended up being used as a vaccine, and Max Tyler passaged the yellow fever SIBI strain 176 times in monkeys. Then he learned monkeys are very expensive, of course, but then he could passage it in embryonated eggs. That is, eggs that had just been fertilized, chicken eggs, and that it would grow in the growing central nervous system of this young embryonated egg. And they could then get those, isolate it from those eggs and develop a vaccine. It was called 17D. It gave you immunity because it was a live yellow fever vaccine, but it had markedly decreased virulence, so you wouldn't get sick. So in his Nobel Prize, Max Taylor said, he acknowledged all those who had gone before him, who gave their lives. He said, those who gave their lives and gained this knowledge were truly martyrs of science. They were. They didn't do it for money. They did it to help humanity, and they ended up giving their lives in so doing. So how's this yellow fever vaccine made? And how it's made? Now, Max Tyler published 3 articles in Journal of Experimental Medicine explaining this. They were kind of back-to-back articles of what got him the Nobel Prize. But basically, when they injected the yellow fever, the 17D strain, into eggs with live chick embryos, they incubated them for 4 days so that the embryos would grow. They cracked the egg open, removed the embryo with its early brain and spinal cord. They just mince up to destroy the embryo in a centrifuge and a blender. And then they freeze the supernatant either on dry ice, or more recently, they lyophilize it and store it as a frozen powder that can be reconstituted and injected into a person. So 1 million chicken embryos are killed each year to make the yellow fever vaccine. This approach was also used for influenza vaccine. It still is, and greater than 100 million chick embryos are killed each year to make influenza vaccine. So if you're a vegetarian, you're adverse to killing animals. I mean, that's an interesting fact, just for the influenza vaccine. So initial yellow fever vaccines contaminated 50,000 US soldiers in World War II with hepatitis B. And the reason for that is that they added human serum in the production of the vaccine, and that human serum they didn't realize had been contaminated with hepatitis B. Because it was World War II, what happened, the Brazilians discovered it, where they did the first yellow fever vaccine trial, because it was endemic in Brazil because of the mosquitoes, and it was working. But the Brazilians were very good. They would come back months later and look at every person, and some of them had liver failure or hepatitis. And they figured it out that it was the human serum that was being added. And they notified America about it. They notified Rockefeller Center in America, but the Americans didn't change what they were doing. They kept using human serum that was contaminated with hepatitis B. And then Later, none of these soldiers, including generals who'd gotten the vaccine, mostly in the Pacific Theater, were ever told its cause of why they got hepatitis B, which, of course, this virus never goes away. It either kills you from acute hepatitis or causes chronic cirrhosis with its liver failure or liver cancer. Well, it also turns out that they were using chicken eggs, that is avian bird eggs, and they were contaminated with avian or bird leukosis virus. It's a virus, ALV, causes leukemia in birds. So basically, there was never an investigation to see what happened if that ended up causing leukemia in humans. But starting in 1967, only eggs from ALV-free hens were used. So if the mother hen had ALV, it turned out that virus was passed in the eggs. And so the developing embryo would have it. And then if you make the vaccine from that, you could pass it on to a human being. But now they only use ALV-free hens. And the other thing they discovered was that yellow fever vaccine, although highly effective in preventing yellow fever, because it's a live attenuated virus, it could be lethal in an immunocompromised person. That's very important. You don't give a live vaccine to someone immunocompromised. And this is where they learned that. And in fact, there are cases in the literature where someone who's giving a kidney to their sibling had been vaccinated for yellow fever just within a few weeks before they donated their kidney, and they passed that yellow fever live attenuated virus to the recipient of the kidney who's on immune suppression to prevent kidney rejection, and they get yellow fever and die. So you cannot have a donor of an organ who has been recently vaccinated with yellow fever vaccine. Otherwise, the recipient will get yellow fever and could quite likely die. So what about poliovirus? This is the last before 1960 of the vaccines. It's a single positive stranded RNA, acts as one big piece of messenger RNA with one reading frame that's cleaved into about 11, 13 proteins. It's transmitted by oral ingestion, and it localizes in the GI Peyer's patches, that is lymphoid aggregates in the ileum, and it sheds in the stool for 60 days. But in some people, it persists forever, shedding poliovirus for up to 28 years. And diseases arises from it infecting motor neurons. Now, normally, even a person, the vast majority of people get polio, don't get poliomyelitis. They get infected with the polio virus, but it can't cross the blood-brain barrier. So there's something that has to allow it to cross the blood-brain barrier to get polioencephalomyelitis with permanent paralysis or death. So how did polio come about? Polio pandemic paradoxically arose from industrialization and cleanliness. So it was human activity that caused polio to become a disease. And it was basically removing sewage from drinking water, which of course took care of like cholera and problems like that, which we think is, so disgusting, sewage in drinking water. But before the industrialization, with rural America, it was very common that the water you drink had sewage in it, either from animals or humans. And women breastfed back then. Well, the baby then would swallow, polio virus, but it was being breastfed. And so antibodies from the mother went into the baby and protected it from getting poliomyelitis disease. But they were exposed to the polio virus, so they developed their own immunity to it. So once they stopped breastfeeding, they were immune to it. So polio basically didn't occur because babies drank dirty water filled with sewage and women were breastfeeding. Once those things changed to the Industrial Revolution, it became a disease. And surprisingly, it was a disease of the wealthy or more or better to do, whose parents weren't breastfeeding them. And people like President Roosevelt, who came from a very, very wealthy family and has developed poliomyelitis. Well, I'd love to keep talking about all these things, but there isn't time. So I'll just have to say, read Kill Switch. You learn about the March of Dimes. The March of Dimes took over trying to find a cure for polio. And it was a volunteer group. And if you asked for help for medical care, they paid for everything. You didn't have to prove you needed the money. Just you asking was enough for them to pay for all your medical bills. They also paid Howe and Bodine at Hopkins to figure out the different subtypes of poliovirus, because that was the first thing they needed to do. If you're going to make a vaccine, you have to know how many types of virus there are to include in the vaccine. And they did that by injecting the vaccine into the brains of monkeys If they survived from polio, if they died, they died. But if they survived, they would inject another type of poliovirus in there and see if they were immune or not. If they weren't immune, that was a different type. Well, they found there were three subtypes of poliovirus, so the vaccine had to have all three. Then the March of Dimes funded John Enders at Harvard, who got the Nobel Prize in 1951 because he figured out how to culture this virus in cells. So you didn't need, you know, animals to do it. You could use cells that would to culture it and grow it in a lab and collect the virus in the supernatant, so you'd have a whole bunch of virus. And again, he did that with measles. He did it with poliovirus. Got the Nobel Prize for it. It's interesting. John Enders, they gave him the money to do it, and for the first time, University Harvard said, "We won't let him take the money to do this unless you pay us for being the administrators, just to have the institution that they turn the light switch on." So they agreed to pay Harvard 26% of whatever they gave Enders. And that started this concept of indirects. Whenever you get a grant, you have to pay the institution a certain part of it. Some institutions now in America mandate 80, 90% indirects, even though all the work and the idea and everything's done by the investigator. Anyway, once they learned how to grow it in culture, they could now have a ready source of the virus to use as a vaccine. And there were several types used: Koprowski, Salk, and Sabin. Koprowski was actually the first vaccine. It was live attenuated, that it has been passaged several times, so it appeared to be less dangerous. They used it in the UK, but the people in the UK did something extra. They collected the stool from the children who got the vaccine and injected monkeys, and found out that many of the monkeys were still getting polio and dying from paralysis. So they realized the virus hadn't been adequately attenuated. They shut it down. Later, Koprowski restarted that study in Uganda, Africa. Very interesting read, kill switch. Salk took a different approach. He took the Pasteur approach, like rabies virus. That is, he killed it. He inactivated it with formalin. And it was very successful. It kind of eliminated polio virus after, the trial was done. It was the biggest trial in America at that time. And, but because it was successful, church bells rang throughout the country, and the society was all behind it. The March of Dimes is called the March of Dimes because to develop money, they passed a hat before every movie that people go to in movie theaters, asking for any change they had to help the March of Dimes, and people would give their change. So it was the Depression. They didn't have much money, but they give their change. So from that, they had the money to do this. That's why it was called the March of Dimes. But anyway, there was some catastrophes with the Cutter incident. I'll let you read about that in the book. And then Sabin has said he felt that a live oral virus would be much better and much cheaper. America had already developed the Salk vaccine. It worked. They didn't want the money or time to do another vaccine. He took it to Russia, where they ran it and showed it worked. That swallowing it one time worked. It was a live attenuated virus. And because it was passed through the feces of whoever took the vaccine, neighbors could get it and also get immunity. And so because it was so much cheaper, Salk had to be injected 3 times, Sabin, you just drank it once. It became the standard in America. People stopped doing Salk and they did Sabin, until they realized it could revert back to its pathogenic virus and cause paralysis. And indeed, over the last 10 years, the only cause of polio-induced paralysis in America was from the Sabin vaccine itself, where people, got it from the drinking water, or they, picked it up from a relative who had been vaccinated with oral live Sabin vaccine. So America switched back to Salk. But because it's cheaper, the rest of the world, especially Asia, Africa, get the Sabin, which is kind of weird because they can fly to America and it's in their stool for Americans to be exposed. Bernice Eddy is an interesting story. She's at the NIH and she was looking into the toxicity of these poliovirus, Salk and Sabin. And she was the one that discovered the Cutter incident, where the Salk vaccine had not been adequately embalmed or killed, and that it was still partially alive, and that many children got polio because of the Salk vaccine from the Cutter company, and several had died. And she reported it before it was released on the population, but the NIH squashed it and didn't let her to continue to work on it, and then it broke out and happened. And so later she continued studying the virus, both Sabin and Salk, and realized when she injected hamsters with it, about 90% developed tumors. So she thought this vaccine caused tumors. The NIH did a epidemiology study about 2,500 people, and they didn't show an association. So they said it occurs in hamsters, but not humans. And that was the end of it, except Bernice Eddy was forever forbidden from studying poliovirus vaccines. That was her reward. Well, if you look at the study where they looked at tumors, most of the people, of course, had the common tumors, breast cancer, lung cancer, things like that. And it doesn't appear to correlate that. But then much later, after 2000, Dr. Carbone, who was at the NIH at the time, began to investigate because the polio vaccine was grown in monkey kidney cells, and monkey kidney cells have a virus called SV40, simian virus, monkey virus 40, and that contaminated the vaccine. That was known, just like avian leukosis virus contaminated the yellow fever vaccine. This monkey virus contaminated the human polio vaccine. But what he found is that virus does cause cancer, but it does it by suppressing anti-oncogenes. So it suppresses genes that prevent cancer. That's what SV40 does. So it's one of the steps in the multi-step process of getting cancer. And it only does it for a few cancers, mesothelioma, sarcomas, lymphomas. And now when you look, there's a markedly increased incidence of SV40 in tumors from mesothelioma and sarcoma and lymphomas, not in the major common solid tumors. So the epidemiology study was wrong because they weren't looking at the right subset of rare cancers where it contributes to tumorigenesis. And that's a caveat to be learned in epidemiology studies. People think it's guilt or innocence. It's not. Epidemiology studies draw associations, and it depends on what you're studying. And if you don't know that it's only this small, rare group of cancers, you're not going to find, you'll think it's innocent of doing it, when in fact it's guilty. You're testing the wrong group of people. So as a result, the FDA now mandates xeno-free, animal-free materials in the development of any product except for vaccines. And the reason that's still an exemption is you need animal cells to make vaccines. So it's an exemption. But now mostly people use like a Vera cell line, which was developed in Japan. It's still a monkey cell line, but it's been checked for every known, that is, quote, known adventitious material or virus, and it's free of it. And, so people use things like that instead of killing live animals to get the cells to make the vaccine. And those animals may be contaminated with all kinds of viruses that could be passed on to the human population. So here's vaccines post-1960. This is the molecular biology age, and it's totally exploded. So now, before 18 months of age, children have around 30 different vaccines given to them. And it's a result of this explosion in molecular biology. So molecular biology changed how we interact with viruses. And of course, the title of the book is The Interaction of Viruses with Humanity and How It Led to COVID-19. So I don't have time to talk about these, but read Kill Switch. I talk about, you have to have a little background in molecular biology. So in the center of the book, I have a thing on atoms, the metals and non-metals and forces, because basically atoms are run by one force, whether it's the electron binding to the proton or the bonds binding atoms together, or enzymatic reactions, or how a cell works, whether a muscle contracts or, whether we can walk, how we interact with each other. All our life that we see around us is controlled by that one force, that is electromagnetism. It's the same thing that runs your computer that you're watching this on right now. It's what runs atoms. And in fact, what is an electron? And, it's probably, there's a beautiful scientific paper, very recent, raises a bunch of questions. It says electrons are just a donut-shaped circle confined of electricity, electromagnetism. The magnetism and electricity are the same things. So it says that's really what they are. They're particles of electromagnetism. Anyway, it talks about DNA and RNA, or genetic continuity. RNA probably preceded DNA because it has an enzymatic effect as well. That is a ribozyme activity. But RNA is 10,000 more times to be mutated easily than DNA. So if you want to preserve your genetic heritage, DNA is better. And that's probably why, as life continued on, the genetic heritage got shifted to DNA, although RNA is still intermediate. Proteins, people think of proteins as scaffolding like collagen or tendons, bones, but or your hair. But proteins walk. I mean, enzymes move to make things happen. Dynin that carries rabies up the microtubule to the nucleus, it walks up there. So that walking, that enzymatic function, that three-dimensional change in structure that allows cleavage or cutting or new bonds to form is from electromagnetism, which is the force of life. It's probably why, these early proteins are what came before the chicken and egg. It's, the force of life itself. I talk about mercury and the occult preservatives. Well, what is a preservative? Preservatives of a germicide, but there's a lot of things about mercury that are super, super interesting to learn there. Aluminum, which is used as an adjuvant, stimulate your immune system. You don't need that with a live virus, but it's often used in vaccines, and we talk about that. And then HIV and AIDS. There's no vaccine for it, but HIV, where it could have arisen, and the drugs now used to treat and control it, turning a acutely lethal disease into a chronic disease. And of course, I bring up the Uganda study, which I think most people aren't aware of, despite the fact it was published in a journal. Read Kill Switch. It's going to really be amazing. I talk about HPV, human papillomavirus, and that's, the first vaccine to prevent a cancer, cervical cancer. Once you have cervical cancer, it doesn't work. There are now attempts to look, and I discussed in that chapter, to make it effective for once the cancer already exists, but it can prevent cervical cancer. And of course, like all vaccines, there's controversy with it. So, but it's the first attempt to use a vaccine to either prevent or stop a cancer. HPV, and you'll find that super interesting. But what I want to come to now is just two more viruses. One is Ebola, which is a filovirus. Philo is Latin for worm or thread-like. And as you can see, it's this long thread-like virus. So it's a single-stranded negative sense RNA of only 7 genes. That's it. And there are 6 types of Ebola virus. It was named for the Ebola River in Congo, Africa. You find a lot of viruses come out of Africa. It's almost like this is the origin of life. Humans supposedly came out of Africa as well. So it causes death through hemorrhage, hypertension, shock, organ failure. It's very lethal, 90% mortality, at least the Zaire strain of Ebola virus is. And that's the second most lethal virus behind rabies. The virus outbreak in 1995 movie called Virus Outbreak by Dustin Hoffman was really about Ebola virus. It's about a monkey got into that with Ebola virus that got into the United States, and their attempt in hazmat suits to quarantine it and prevent spread. Well, it was discovered in 1976 in the blood of Sister Miriam, a nurse in the Congo. And how that came about and spread among people is that the nurses were given 5 needles a day in 1976 to reuse over and over on patients. They would wash them between each patient. They wouldn't sterilize them. So if a patient had hepatitis or Ebola or whatever, HIV, it could be passed to the next patient. And it's just they didn't have the resources or money to buy reusable syringes and needles like in America. Even in 1976, they didn't have that. So the vaccine for Ebola virus is a recombinant virus that never existed before on this planet. So what happens is it was invented by John Rose at Yale, and then it was taken up to develop first in Canada and then later by Newlink. And basically you take a vesicular stomatitis virus that's not pathogenic to humans. It causes vesicles in hoofed animals. Very, very rarely it'll cause a vesicle in a human, or super rarely it'll cause encephalomyelitis in a human being. But for the vast majority of humans, it does nothing. It's not a pathogen. So here it is. It's actually of the same species as rabies. It's a bullet-shaped virus. It only has these 5 genes. They remove the glycoprotein, which is the outside that allows it to bind to cells from vesicular stomatitis virus. They remove it, and so you have what's left. These are the genes. So if you want to look them up, they're right here on the right-hand side. And then in that, they would insert the glycoprotein, or what's on the binding protein of the Ebola virus, in there. So now you have this recombinant new virus that's never existed on the planet before. It is a live virus, but it will give you an immune response to the binding protein on Ebola virus, but it doesn't carry the pathogens Ebola does. It carries vesicular stomatitis viruses that allows it to live and produce, but it doesn't cause disease in humans, just like VSV doesn't. And so they started injecting people with this and found that it was very effective in preventing Ebola virus, and it stopped that pandemic. But as a result of something that was so great in stopping such a lethal disease, and again, now there's another outbreak of Ebola, it's a different strain. And the new strain of Ebola that they're talking about in the news today is resistant to this vaccine, which was developed for the Zaire strain. The new strain of Ebola virus is about 30 to 50% mortality, which again is less than 90% of the Zaire strain, but that's still a very significant mortality, and they don't have a vaccine for it. But in any event, it's a new strain. But this that was so effective against the most lethal strain, we, as a society, we crossed the river Rubicon. That is, we use molecular biology to create new virus, a new life that has never existed on this planet before. And when you change life, there's this attitude that it's an algebraic equation. You change one side and you get an equivalent change on the other side. And that's not true. A life is chaos theory. That is, like a butterfly flapping its wings on one side of the world could have very late fractal and unintended consequences decades later, like a hurricane on the other side of the world. So, this was a great success. The risk benefit was well worth doing. It's a highly effective vaccine. It's a live vaccine. You don't need an adjuvant, but it's a whole new life form. And you got to remember, we are descendants of viruses. Our foundation rests on the life of viruses. So, could bad things happen taking this approach? Well, that brings us to coronavirus and COVID-19, which is a coronavirus abbreviated COV. It stands for crown. And you can see the electron micrograph picture of the coronavirus. It's like this crown sticking out of the side of it. That's why it's called a coronavirus. It's a single positive strand RNA virus with 10 to 15 open reading frames that produce about 30 different proteins. Well, there's hundreds of coronaviruses naturally, different strains found in about 50 animals, with bats being the big natural reservoir of coronavirus. There are two theories that exist for the origin of COVID-19. COVID-19 is also known as SARS-CoV-2, COVID for coronavirus 2. And those theories are an animal source versus a lab leak. And if you were at all here during the COVID-19 pandemic, especially for the first 2 years, there was a lot of back and forth acrimony about whether it was an animal source or a lab leak. And I think getting to the bottom of this is really important, not to prevent vaccines or a kill switch, but to prevent complications from a vaccine, and that perhaps we need kill switches on vaccines as well. So, evidence in favor of COVID-19 being an animal source. So, COVID infections have always been zoonotic, that is, from animal to man. They always have been. There's 4 of the many different coronaviruses that can cross from animals to humans to cause common cold. That's well known, but it's just a common cold. And, people aren't too worried about it. There was one coronavirus called SARS, COVID-1, that caused lethal epidemic that came from animals. And that occurred from animals bought in Chinese wet markets in 2002 to 2003. Now, what's a wet market? Basically, you go to a food market where they have live animals. You pick the animal you want. They cut its head off or its throat. It bleeds out. And they give you the animal. You take it home. You cook it up very fresh. All kinds of animals, birds, reptiles, everything. And so it's very fresh meat, but there gets to be blood all over the floor of the market. So they just wash it off. So they're called wet markets for that reason. But anyway, it was discovered to come from the wet market from animals. And once they had euthanized all these animals, the epidemic in the early 2000s stopped. It never became a pandemic. So there's another COVID virus, Middle East Respiratory Syndrome coronavirus called MERS, that crossed from bats to camels and then to humans and caused a lethal epidemic, but not a pandemic in 2019. So it seemed very likely that COVID-19 probably came from an animal as well. And in fact, there was a paper, and it's called the Proximal Origin Paper. They studied the genetic sequence of SARS-CoV-2, that is the virus that causes COVID-19. It's a coronavirus. And it was reported in 2020 in Nature Medicine by Anderson and all at Scripps. And there were a bunch of unnamed contributors to that paper whose name are not on that paper. That's very important. And that information came out by a Freedom of Information Act that the Congress had to sue to get released and to get the emails to find out what happened. So, but anyway, in that paper, they conclude, we do not believe any type of laboratory scenario is possible. And they say that because no laboratory sequences reported by the Wuhan lab in China, which was funded by the NIH, were sufficiently equivalent to COVID-19, SARS-CoV-2. But if you read that grant, and I have it, and I've read it as well as other grants, what it says is they will not release the sequence of any new coronavirus they've made until it's published in an academic journal. And the NIH agreed to pay for that. So just because they hadn't released it yet doesn't mean they hadn't made it. So what's the evidence in favor of a lab leak? And the history is that the NIH, NIAID have been funding gain of function already in influenza virus, another respiratory virus. COVID is a respiratory virus, influenza is a respiratory virus. Well, what is gain of function? Gain of function is when you take a virus that's not pathogenic to humans and you molecularly change it, use molecular biology to change it so that it infects and can kill a human being. That's gain of function. And that was being done in influenza, funded by NIAID at NIH. So how do I know that? Because it was done in the Netherlands in 2012, the lead author being Fournier, and that was it. What they published the results, and they published it in Science that they had genetically engineered influenza H5N1 to easily spread among ferrets. And that, if it spreads easily among ferrets, that's the animal model that it'll spread easily among humans. That's well known for people that work in influenza. Well, H5N1 had short epidemics, never became pandemics, just short epidemics because it could infect people, but it could not spread easily. So it couldn't make a pandemic, so it would fizzle out. But they actually changed this virus so it could spread easily, and they reported it in Science. They made this a whole new virus that had never existed before. It's little derived from H5N1. So that resulted in an ethical uproar, and this was published in many different journals. Should gain of function even be allowed? Should publications of gain of function be allowed? Can scientists regulate themselves? So as a result of this uproar, the Netherlands voluntarily shut down their gain of function on COVID for 60 days, but they restarted it afterwards. And 60 days is an inadequate period of time to assess it and put in safety mechanisms and kill switches to prevent bad things from happening. But it quieted the media down, and that was that. So what happened then is, what's the NIH attitude towards gain of function? It funded it. Well, Fauci wrote a paper, saying how great it is that they voluntarily suspended this for 60 days. But he went on to write, he wrote it. This is in publication form. He actually wrote it. He's the only author. It was published in Molecular Biology in 2012. And he wrote, these are his exact words: in the unlikely but conceivable turn of events, what if a scientist becomes infected with the man-made virus, which triggers a pandemic? Scientists in the field might say, as I have said, the benefits of such experiments and knowledge outweigh the risks of a pandemic. So that was the attitude. And that was just not the attitude of Fauci. It was the entire NIH, because nobody stood up and said, well, the risk of a pandemic, what are you talking about? A death of one person, 100 people, 1,000 people, 1,000,000 people, 1,000,000,000 people? Is it worth it? Now, I'm all for science for knowledge's sake, because a lot of times decades go by before you understand what you've really learned to help humanity. But there was a cavalier attitude to doing this. In his own words, he put it in publication. So they've been funding it in influenza. Well, it turns out they funded it in coronavirus as well, gain of function. So in 2015, funded by NIAID at the NIH, Burt at the University of North Carolina, and Shi in Wuhan, China, genetically manufactured an entirely new bat coronavirus that can infect and damage human epithelial cells. And they published that in Nature Medicine in 2015. And some people will argue about the definition of gain of function. In fact, that has happened in the press, but they themselves, in their paper, if you read it, they called it gain of function because it was. They took a coronavirus that couldn't bind to human cells and they changed the receptor binding domain on the spike protein and it bound to human cells. And they published that. Nature Medicine, and it clearly says who funded it. So Nature Medicine put up an editorial, and the editorial was titled Engineered Bat Coronaviruses Stirs Debate Over Risky Research. You take a virus that's not pathogenic, you make it pathogenic to humans. That's risky. So this gain of function was defended not just by the authors of the paper, that is Burt and others, she in China and at University of North Carolina, but by Dasiak at Echo Health Alliance in New York. He wrote an editorial to the editorial saying, oh, this is very important, for research. So due to safety concerns, the White House Office of Science and Technology Policy under President Obama in 2014 stopped all gain of function on respiratory viruses. That included influenza and coronavirus. They looked at this and said, this is risky, and they stopped it. And the way they said it's risky is in the United States, they had on average 3 complaints of safety concerns every week on labs working with viruses. And because you're supposed to report them, they said, this is way too much. So they just shut gain of function down. And it turned out at that time, over 14 universities were being funded by NIH, NIAID for gain of function research on viruses. Again, this was under the Obama administration. So what did the NIH do? What did NIAID, the institute headed by Fauci, do? They continued funding gain of function research, but they couldn't legally do it in America. So beginning in 2005, they funded it in Wuhan, China, by sending NIH money via an intermediary, Dasiak's Echo Health Alliance in New York, to Wuhan, China. And they collaborated back with just scientific information from the University of North Carolina. But in the private world, if you circumvent money for an illegal activity by sending it through a third party, that's called money laundering. But yet it was totally legal to do it because they weren't doing it in America. But they continued doing it despite the Office of Science and Technology policy saying you can't do it anymore. And they did. And this is their grant number. You can look it up. So people involved in coronavirus gain of function and in the proximal origin paper that said it could not have come from Wuhan, China, were conflicted. They should not be the judge because they were conflicted. They had an interest in seeing that this is not arising from a lab. So if we look at University of North Carolina, Burt, so these are kind of triangles within triangles because this thing, to follow it, becomes very complicated and you have to pay attention. But these are all people that work together on, either the proximal origin paper or on gain of function on coronavirus. And in 2015, University of North Carolina reported in Nature Medicine that they'd done gain of function on the receptor binding domain. It can infect human cells. But then, despite the Office of Science and Technology Policy from the White House saying you can't do it anymore, they continued to fund it in China with Xi, who was one of the original collaborators. And of course, in China, she was involved in that original 2015 paper in Nature Medicine, but she continued gain of function after it was shut down in America by an NIAID grant that went through Dasiak Echo Health Alliance in New York, and also helped in collaborating with University of North Carolina, despite the stop on doing gain of function research. So Dasiak himself, he's actually a zoologist, a British animal doctor who ended up heading this big viral research, Echo Health Alliance, and he collaborated on NIAID funding after 2015 with Xi in Wuhan, China, and Burt University of North Carolina after it had been stopped by the White House. So Fauci himself he used to control $6 billion in US funding every year. He got $6 billion. It's an extremely powerful person. And he's well known for his work in autoimmune disease and viruses and vaccines, and has a lot of appropriate recognition for doing that work. But he funded the original gain of function reported in 2014. He funded over 14 universities involved in gain of function, and he funded ongoing, despite the prohibition by the White House, gain of function in Wuhan, China. And it turns out he was a secret author of the proximal origin paper out of scripts by Anderson saying this could not have come from the Wuhan lab. How do we know he's a secret author? His name's not on the article. But we know because the Congress sued the NIH to release emails, and there's a ton of emails back and forth where he advised and was very supportive. And Anderson himself originally said this had to have come from Wuhan, China. If you've seen his personal emails, it was man-made. But they convinced him to switch and say it couldn't have been man-made in Wuhan. You can read Kill Switch to figure out how that happens. So that, Anderson is the principal author of the proximal origin paper, which Fauci got up. I saw him do it, saying, look, here's a paper by these other people, we have nothing to do with, and it says this was not man-made in Wuhan. So the Erasmus Medical Center, they did gain of function in H5N1. They were secret authors of the proximal origin paper. It's all in the emails that had to be legally forced out of the hands of the NIH of how they coordinated and wrote the paper together, even though their names aren't on it. With Anderson, who was the World Health Organization, he supports gain of function. He wrote a book, Spike, and he actually talks how they worked together and coordinated to write a proximal origin paper. They were so oblivious to their bias and their inability then to be objective, because they were big supporters of gain of function, that they co-authored reasons why and had to convince Anderson that it didn't come from Wuhan, China, which they were successful in doing through their emails that were never put as authors on this paper. So after the proximal origin paper publication, which looked at the genetic sequence of COVID-19, the virus that causes COVID-19, the NIH director, Dr. Collins, and NIAID director, Dr. Fauci, and the entire media press echoed that a lab origin is a conspiracy theory. And if you stood up and said, it came out of the Wuhan lab, you were labeled some sort of wacko, a conspiracy theorist. But if you look, two Nobel laureates, Luc Montagnier, who was the first to discover the cause of AIDS, that is the HIV virus, got a Nobel Prize for that, is at the Pasteur Institute, where they made the Pasteur vaccine, a dead virus for rabies, a long time ago. And David Baltimore, who was the president of California Institute of Technology, a really bright Nobel laureate molecular biologist, and the Department of Defense and the US Congress concluded that COVID-19 was man-made in a lab. So they had exactly the opposite conclusions of the NIH that funded COVID-19 gain of function. So what is going on here? I mean, how can this be? Let's look at the genetic sequence of COVID-19. Because again, it was the POP paper looking at the genetic sequence that said, it couldn't have come from a lab. So here's COVID-19. These are the proteins, or the genes, if you will, at the very top. And what we want to focus in on is this S protein or gene. And here it is, called the spike protein. And it's what binds the virus to a cell, either to a human or a non-human cell. And there's two key points in this protein out of this virus. And those points are the amino acids for the receptor binding domain, and amino acids for furin cleavage site. Those are the two key points. And because of those 2 points, it's why initially in an email to Fauci and others, Anderson said this was made in a lab in China, despite their paper would have finally come out saying something totally different. So what we have here is that the receptor binding domain causes the infection. It can cause an epidemic. And that we know was made by North Carolina Barrett, by Wu Hanxi in 2015. They reported in Nature Medicine. After 2015, they continued gain of function in Wuhan under grants by Daisy and Barrick, funded by Fauci's NIAID. So they made this particular amino acid sequence and inserted it so it would bind to human cells. Now, the furin cleavage site here in the binding protein, the spike protein, that allows easy transmission of the disease, which causes a pandemic. So what's important to understand, infection is different than easy transmissibility for respiratory viruses. Infection can cause an epidemic, but then it fizzles out because it's not easily transmitted. Whereas easy transmission, which arises from a different area, causes a pandemic. So why is that? Well, furin is a protein in human cells. And if you have the right furin cleavage site in this coronavirus, they had already put in the right receptor binding domain so it would bind and infect human cells. If you put in the furin cleavage site, as it goes inside the human cell, furin will cleave it and open up the virus so that it can be very rapidly transcribed into new virus particles. So the transmissibility markedly increases. So it's much more infectious and you get a pandemic. That's what the furin cleavage site does. So was the furin cleavage site made by this group after 2015? That's the question, because COVID-19 has the optimal receptor binding domain, which they already published they made. It also has the optimal furin cleavage site that coronaviruses of this type do not ever carry. So did they put that in there? That's the question. Well, it turns out that Dasiak at EcoHealth in New York, again, this was after gain of function was shut down by the White House Office of Science and Technology Policy, and in New York, and that she in Wuhan, China, and the University of North Carolina in Burt submitted a grant to the Department of Defense in 2018 while gain of function was still shut down, 3 years after they published that they had established the receptor binding domain with optimal binding to human cells to infect human cells. In this grant, I've read it, they said they wanted to create the furin cleavage site in that coronavirus. So now it could be rapidly transmitted, not just infect a human cell, but rapidly transmitted to other human cells. The grant, here's the number of it, was called Diffuse, for diffusing the threat of bat-borne coronavirus. It states in the grants, these are their own words, we will introduce appropriate human-specific cleavage sites, that is furin cleavage site, and evaluated coronavirus growth and spread. So it turns out the Department of Defense, unlike the NIH, who was funding the current research, refused to fund it because the NIH said it's illegal gain of function. And that was illegal by the Office of Science Technology Policy. So they refused to fund it. Now, there may have been other reasons they did, and they wanted it funded in China like NIH was doing. But of course, a big part would be taken out by EcoHealth in New York. But the DoD, Department of Defense, said, we won't fund this. And that's the reason they gave. That's the predominant reason. So in 2020, this broke out, this virus in 2019. But in 2020, at the end of 2019, Major Joseph Murray of the U.S. Supreme Court's War Fighting Laboratory found this shelved diffuse grant right here and wrote a summary to the Inspector General of the Department of Defense. What he wrote is, and basically it's what the grant says, if you can read molecular biology, you can read this grant. The 2018 diffuse grant was a roadmap of combining the optimal receptor binding domain and furin cleavage site that was found in COVID-19, that is present in COVID-19 and not in other coronaviruses of this type. So here's the problem. DoD didn't fund it. So the question is, did they still do it? Well, the catch-22 of getting a grant is you need data to get the grant, but you need a grant to get data. That's always the catch-22. So what big labs do, and science, I know, I'm guilty of this too, everybody is, if they're honest, is how do they get around this catch-22? If they have a grant funding, they'll use some of the people involved and reagents and stuff that they have for the funding grant to get data for the next grant. That's how they get the data, because you got to have data to get a grant, but you got to have a grant to get data. That's what they do. So the question is, even though the DoD didn't fund them, did they go ahead and do it? Did they add the furin cleavage site to a genetically modified coronavirus that contained the optimal receptor binding domain that they already put in publication that they had inserted? Well, we don't know the answer to that because there's never been a formal investigation. So defenders of the natural or animal origin of COVID-19 are the NIH, Fauci, and the people whose research NIH funded. Dasiak, Anderson, Farrar, Fourier, Barrick, and Shi, and the proximal origin paper in Nature Medicine. They say COVID-19 arose naturally. Collins and Fauci in the US and British press stated that a lab leak is a conspiracy theory. Defenders of the lab leak as the likely cause for COVID-19. Two Nobel laureates, as I mentioned, David Baltimore, Luc Montagnier, the Department of Defense, the U.S. Congress, and the director of the Centers for Disease Control and the Lancet Commission on COVID-19 have concluded that it was man-made in a lab leak. So CDC Director Robert Redfield stated, I think there's a real possibility that this virus first place was Chapel Hill. That is Barrick at University of North Carolina. So the Lancet, the publication, The Lancet, they actually commissioned Professor Cohen at Columbia University to chair their commission to determine the origin of COVID-19 due to the great controversy out there. They did the right thing. They said, let's get an independent person. It has no bones, in this melee to figure out where it came from. And so Professor Cohen and another professor at Columbia published in Proceedings of the National Academy of Science, very prestigious science journal in 2002, that it likely arose from the Wuhan lab after 2 years of investigation. Professor Cohen himself said, I am pretty convinced that COVID-19, came out of a U.S. lab, not nature. Well, why did they do that? One of the reasons they brought that out in their publication, PNAS, is the furin cleavage site that is in COVID-19 that causes the rapid transmissibility. They'd already inserted the proper receptor binding domain so it would bind to human cells, cause infection. But the furin cleavage site, that could have been engineered to put in there, that furin cleavage site was already being used and had been developed at Chapel Hill, the collaborating institution with Daisiac EcoHealth and Wuhan, Dr. Shi in China. So the question is, did they send that to them? They'd already made it. Did they send it to them? There's never been an investigation to answer that. But he raised that question by putting it in Proceedings of the National Academy of Science. To this day, 6 years later, no animal source has ever been identified for COVID-19. So how could this happen? I mean, I've given you the information. It's your decision to decide what happened, but how could this happen? So Kill Switch gives an answer by explaining the psychology of game theory in the last chapter and the problem with well-meaning laws gone wrong, like the Bayh-Dole Act, and the flow of money in political action committees and drug company advertising. To understand that, you're just going to have to read Kill Switch. If you don't even know what a political action committee is or what's going on there, read Kill Switch and how this can influence what's gone wrong. So people, you and I, are not allowed to investigate ourselves. Well, city, county, state, and federal agencies, departments, and institutions, including the NIH and police, routinely investigate themselves. That's totally appropriate or considered appropriate. So police, when people would say there was police brutality or they were inappropriate, it was a police word against the civilian. And so people always had to trust the police. But now with video cams and everybody videoing the police, something bad happens, there's a report, and the police say, hey, there's no problem here, nothing happened, move on. But eventually the video cam is released, there's an uproar from the general public, and then an outside investigation, and justice is done. But that happens not just in police, every type of government, including the NIH, that happens. Outside investigation is a kill switch and something that doesn't exist in government circles. You and I cannot investigate ourselves for a crime. That's recognized as a bias. But a department, institute, or agency of the government can and does do it all the time. Investigate themselves. Freedom of speech is a kill switch. And remember, again, freedom of speech was being stifled. Calling someone a conspiracy nut stifles freedom of speech. Consent is the basic respect for a person, and it is the final and most important kill switch. Not coerced consent, where you lose your job or you can't get in an airplane or you can't work if you don't get a vaccine. Honest consent. That was forgotten about during this push for vaccinations for COVID-19, whether good or bad. Consent is respect for person. And if you don't have it, you don't respect people. Kill switches prevent good intentions from going bad. They not only protect the society or the person, they protect the investigator. And that's the important thing. And that's why we must always have outside investigation, freedom of speech, and consent as a physician when you deal with a patient, and respect their decision. It's their life, it's their body. If the vaccine works, the person who gets it has no fear if that person doesn't get the vaccine, if that other person doesn't. So, what I want to say is gain of function continues to this day. It's still ongoing. It's now given other euphemisms like dual function research. They just changed the name, but it's the same thing. Why not insert a biologic kill switch when you make a new virus that never existed on this planet? It can be done. I did it a long time ago, like 2 decades ago, published a paper on it. It can be done, but it's not considered. But why not do that if it escapes the lab so you can stop it? You can make, I'm not against making new pathogenic viruses for knowledge and knowledge sakes that, decades later may come out to help people. But I very much am concerned about the risks of making these new pathogenic viruses that have never existed on this planet. And I don't think, despite the giant red flag and 8 to 10 million dead from COVID-19 and all the lives disrupted and problems that arose with failed businesses and divorces and everything, I don't think we've learned the lesson because it's still ongoing. So we need to take a look at that. And so I'll just, when you read Kill Switch, you'll get a better understanding of this if you're confused. And so let me end by saying the war between viruses and humans will never end because we have met the enemy and he is us. Thank you.